Okay, hello grade 9. This is lesson number 7 of our unit on evolution, factors affecting gene pools. This lesson will not be on your quiz. So if you're in my class and you're a little behind because of my situation this week, then you can still do this lesson so you're not going to get behind for next week, but it will not be on your quiz. Okay? So here's your tell for today. It's 2-pager. The first page is for notes, and the second page is for 2 case studies you're going to analyze. So what is genetic drift, and the founder effect with the diagram, and bottleneck effect with the diagram. And the 2 case studies, elephant seals and the Pennsylvania Amish. So pause the video and draw your template, please. Okay, so first things first, what is genetic drift? So if we're going to talk about genetic drift, you need to remember, keep in the forefront of your mind, what a gene pool is, and remembering that the gene pool is the complete genetic variation. So all the different alleles in a population. How many different alleles are there? How frequent are they? Is it mostly one allele, a little bit of variation, or is it fairly even mix? That situation is the gene pool. So in this diagram here, here's the gene pool. As the original population here, we have a frequency of 0.25 for each of the 4 different alleles. So whatever trait this is, let's say this trait is leg length, there's 4 different versions of that exist, okay, in that particular population. So we quantify this, we give a number to it with allele frequency. So just say, look at the numbers here, look in variety, we have the actual number, 0.25 for each one in that situation. And last class we talked about the fact that if the allele frequency changes significantly, if it's changing, then we see the population is evolving, that the population is undergoing natural selection. And that is almost always the case. But every once in a while, something random happens, something completely by chance. And that is called genetic drift. Genetic drift is when the allele frequency changes the population, but it's caused by chance. Some sort of change in the population that is completely happening without any selection. It's not selected for because one is better adapted. It's just completely chance which organisms are going to survive and which ones are not. So look at this image here. We have our original population with 4 alleles, 0.25 frequency for each. And then some sort of chance event happens. Maybe it's a natural disaster. Something happens, and of this whole population, only this very small number of individuals actually survived. Okay, so that's 5 blue, 2 pink, and 3 green alleles, and not a single yellow allele survived. This has nothing to do with fitness, which one is better able to adapt it. It's just about chance. So maybe something happened, maybe a volcano erupted, and these particular ones are the ones that happened to survive. Maybe they happened to be luckily in the right spot and didn't get hit by any flying debris or anything. Something like this. New frequencies of the alleles are now 0.5 for the blue, 0 for yellow. Yellow is gone completely from that population. 0.2 for pink and 0.3 for green. This is very different from our original allele population here, allele frequencies. Then over time, after that event, that population will start to will grow again based on the limiting factors available in the ecosystem, and we have this new population that's going to have fairly similar frequencies to what we had in the after chance event. So you have 0.45 of blue, 0 yellow, of course, they're all gone, 0.25 of pink, and 0.3 of green. So even though the population increases in number over time, the yellow allele remains gone, and the blue is the most common one. Now this can then undergo natural selection, and this can change, but the idea is this chance event completely changed the allele frequency. So the important thing here is a chance event. This is not caused by selection. There's nothing happening as far as what's better adapted. It's just a chance event. Okay. So here's a really good picture of this. I like this cartoon that the Amoeba sisters came up with, where we have genetic drift. You're going to get this little frog as a result, but it's completely due to luck. It has nothing to do with being better adapted to the environment. It's just lucky you happen to be the one that survived or moved to a new environment, whatever it happens to be. Whereas in natural selection, you have the ninja frog here who's really tough and better adapted and survived because they're the most fit, and that's why the allele frequency changed. Why this became more common in natural selection, and this could become more common in genetic drift. Chance, selection. 2 different things that both can cause a change in the gene pool, a change in allele frequency. Okay, so the first one here is the founder effect. So in the founder effect, maybe you're going to have some members leaving these original environments. So here's my original population, and maybe these four ladybugs and these four ladybugs choose to immigrate to a new environment. These ones fly away together to a new place and found a new population. And these ones go to a new place and found a new population. So they're moving to a new island, maybe they're moving across a river and then the river floods, they can't get back, or something happens that's going to isolate them in a new environment. And only those founding members are going to create that new population. So having all these different colors of ladybug, in this new population, it's only these two colors, really three, that are available. So we have this very different population looking here than we would here. Same thing here. So we have none of the brown ones are present. So we have no brown ones in the eventual descendant population. And here we have two brown ones, so we have a bunch of brown ones being present. This is a lot less diverse. The diversity here is a lot less than here. Okay, because I only have these four ladybugs to make this new population, so we lost a lot of our genetic diversity. Different allele frequency is going to happen here. And a big part of this is because we have a small population, they're going to be inbreeding. These are going to breed together to produce these descendants, and that can reduce the variation. Some examples of this, in the Afrikaner population in South Africa, there's a high incidence of Huntington's disease, which is a genetic disease that affects your brain, normally later in life. And this is just because the population that went from, I believe it was Holland to South Africa. The Afrikaners there, they stayed in a very small population and there was interbreeding for many generations. They founded a new population and they didn't mix the locals, so it had a very limited genetic diversity. Only the alleles that were brought with them made that new population. And that happened to have slightly more frequency, more alleles for Huntington's disease than others, and there was a higher incidence of that in that particular population. Another example is in the fundamentalist church, mostly in the United States. There are a lot of people that have what's called fumarase deficiency. And fumarase is an enzyme that you need for cellular respiration to get energy in your mitochondria. So people in that church are encouraged to marry within the church. They're encouraged to have one husband and multiple wives. And it's a rather small population, so we have quite a bit of inbreeding happening there. And then we get this higher frequency of this deficiency. These are both recessive traits. They don't show up very often in the main population because people have one dominant, excuse me, one dominant trait to kind of mask it. So founder effect, you have less alleles founding to make the sense that they're going to have a different population frequency of alleles, different number of alleles present here. So in the Afrikaner, maybe we have 4 Afrikaners making the new population, was more than that, and in that sample there happened to be more people who had or were carriers for Huntington's disease, and that got passed on to more frequency in the descendants. Same thing with the fundamentalist church. Okay, the second one is called the founder effect. Oh my gosh. This is your drawing for the founder effect. Make sure you draw this one and use color to help you show the different traits. You need to use color, guys. And the next one is the bottleneck effect. This is also the drawing you have to draw for this one. And again, use color. So this is when we have some random event is going to cause a sudden and severe decrease in population size. Look at that. Typo. Okay, so we decrease in population size, and then if that population is able to recover, the allele frequency will change a lot. And it's called the Bottleneck Effect because if you take a look at the image here you need to draw, is this is my whole population, and let's say this event here is a big overhunting thing happening, then only a few will survive. And you think about bottlenecks happen in lines, a lot of bottlenecks happening, you're waiting to get into a concert, there's a whole crowd, the doors open, there's only 2 doors, and it slows everybody down. Not everyone can fit through those doors at the same time. That's the bottleneck. That's the narrow opening that reduces it. If this bottle was fully open here and it's the same size everywhere, a lot more would pop out. So from this giant original population, only very few survive the bottlenecking event. A good example is overhunting. And then the whole new population, if it's able to grow, will grow from that surviving population. So you look at the frequency of alleles here, and this one is a pretty good mix of yellow and blue. but in this one I have a lot more blue than yellow. That's a bottleneck effect. Examples, right whales were whaled almost to extinction, and right now the populations that exist have a severe bottleneck effect and a lot less genetic diversity in that population than there would be otherwise, which is not good for them for evolution. So if something happens to them, like climate change affects them really severely, they're less likely to be able to adapt because they have less variation in the population. Cheetahs experienced a natural kind of bottleneck, and during the last ice age, a lot of cheetahs died, and the population that is left is very closely related to each other. And so now when people are breeding cheetahs in zoos and conservation programs, they need to really carefully track the relationships and how closely related two individual cheetahs are to avoid inbreeding and try and get that genetic diversity back up again. Okay, that's the bottleneck effect, second example of genetic drift. Okay, so to test yourself, I want you to compare and contrast the bottleneck and the founder effect. There's 4 similarities and 1 difference. So take a minute and do a compare and contrast. Okay, so hopefully you found 4 similarities would be that they are both are followed by genetic drift, which results in changes in allele frequency. So they both result in changed allele frequencies. We have initially a loss of genetic diversity. Both have a small number of individuals that breed each other, and that could involve inbreeding, not always, but it could, depending on how small the number is. And that's going to result in a new population which carries alleles that are unlikely to be a true representation of the original group. So for example, Huntington's disease is not very common in the population of humans as a whole on this planet, but it's much more common in African population in South Africa. The difference is what causes it. It's the event. So individuals are killed, reducing the choice of mates in the bottleneck effect. But the founder effect is are ecologically separated. Maybe they move to a new place. For some reason, they can no longer interbreed. Okay, so make you double check you understand it. Use this to help you do that. Okay, so the last part of today's lesson and your assignment, which I recommend you work with a partner on, is to analyze 2 examples of this, 2 case studies. One is the Pennsylvania Amish down here, and one are the elephant seals. So I've uploaded a document that has links to reliable sources to click the links, read them, and summarize the key ideas for each of these examples. So how are these examples of genetic drift? Oh my gosh, so many typos today. And how and why the allele frequencies changed. Okay, make sure that you are using unit vocabulary. Okay, it's the toughest part of biology. It's all the vocabulary, so make sure you use it when you're working on this. And I recommend you work with a partner. Okay, discuss it together, talk it out. We can't do that. We're not together in class on campus to do this, so you have to kind of force yourself to recreate it with a classmate. It can be from either grade 9 class, but talk about the examples here. Okay, so that's your homework, is to upload your notes from today's lesson. It'll include your case study analysis. Our quiz 9.2, your quiz will be on Monday, so make sure your notebook's really organized because you have to, you can use it during the quiz, but you won't have time to search through things endlessly, okay? All right, guys, I hope you have a good day, and I will see you 9.2 tomorrow afternoon for our Zoom.